The year is 1900. The Industrial Revolution has transformed societies, bringing forth incredible inventions and advancements. Yet, in the shadows of this progress lurks a constant threat: infectious diseases. Life expectancy was significantly shorter. Diseases like pneumonia, tuberculosis, and sepsis were death sentences. Doctors, armed with limited tools, often found themselves helpless against these invisible foes. They relied on rudimentary methods like isolation and wound cleaning, but often these efforts proved futile. Imagine a world where a simple scratch could lead to a life-threatening infection. The discovery of penicillin would change everything. Infectious diseases caused by bacteria and other microbes were the leading cause of death worldwide. These unseen adversaries wreaked havoc on populations. The Spanish flu pandemic of 1918 claimed millions of lives. It highlighted the urgent need for effective treatments. Parents worried for their children, vulnerable to diseases like polio and whooping cough. Every scraped knee, every cough carried the potential for disaster. Scientists and physicians around the world were engaged in a race against time. They toiled tirelessly in their laboratories, desperately searching for a cure. Amidst the darkness, a glimmer of hope emerged. Scientists like Paul Ehrlich, a German physician and researcher, dared to dream of a magic bullet. Ehrlich's work on salvarsan, a drug effective against syphilis, fueled the belief that a cure for bacterial infections was within reach. His success marked a turning point in medical history. The search for a magic bullet intensified. Little did they know that the answer lay hidden in plain sight, waiting to be discovered by Alexander Fleming. His accidental discovery of penicillin would change the course of medicine forever. Alexander Fleming was born in 1881 on a farm in rural Scotland. He was a curious and observant child, drawn to the natural world. Despite financial constraints, Fleming's family encouraged his education. He excelled in his studies, showing a particular aptitude for science and mathematics. After completing his primary education, Fleming moved to London to pursue further studies. His time at the Royal Polytechnic Institution solidified his passion for science. From medicine to microbiology, Fleming's journey into the world of the unseen. After graduating from the Polytechnic, Fleming chose a career in medicine. In 1901, he enrolled at St. Mary's Hospital Medical School. Medical school was rigorous, but Fleming thrived. During his clinical rotations, he witnessed the devastating effects of infectious diseases. This experience fueled his desire to find new ways to combat these deadly illnesses. Fleming found himself particularly drawn to bacteriology. Little did he know that this fascination would lead him to one of the most significant discoveries in medical history. The Great War and its impact shaping Fleming's research path. In 1914, the world was plunged into the chaos of World War I. Fleming enlisted in the Royal Army Medical Corps, trading the confines of the laboratory for the battlefields of France. The horrors of war and the countless lives lost to infection profoundly impacted Fleming. As a battlefield doctor, he faced the grim reality of wound infection. The war served as a catalyst for Fleming's research, intensifying his drive to find a solution to bacterial infection. Back to the lab, the quest for a powerful antiseptic. The year is 1918, World War I ends. Alexander Fleming, deeply affected by his wartime experiences, returns to his research at St. Mary's Hospital Medical School in London. Fleming resumes his search for an effective antiseptic. He remembers the countless soldiers lost to infection. The existing antiseptics are harsh, often harming the body's own cells. Fleming envisions a different approach, one that targets bacteria while leaving healthy tissue unharmed. His laboratory is filled with petri dishes, microscopes, and test tubes. Fleming spends countless hours here, meticulously growing and studying bacteria. He experiments with different substances, testing their effectiveness against various strains of bacteria. His keen observation and meticulous record-keeping become invaluable tools in his research. Fleming's reputation as a brilliant and dedicated researcher grows. Yet despite his best efforts, the elusive magic bullet remains out of reach. Little does he know that fate has a serendipitous event in store. An unexpected finding: the mold that stopped the staph. It's September 1928. Fleming returns to his lab after a brief vacation. He begins sorting through petri dishes containing colonies of Staphylococcus aureus. One particular dish catches his eye. It's covered in a strange mold. Fleming notices something peculiar. The area around the mold is clear. The Staphylococcus aureus colonies have been inhibited. Intrigued, Fleming examines the dish more closely. He identifies it as a species of Penicillium. Fleming names the substance produced by the mold penicillin. Identifying the mystery mold Penicillium notatum emerges. Fleming meticulously documents his findings. He identifies the specific species of Penicillium responsible for producing penicillin, Penicillium notatum. He studies the mold under a microscope, observing its structure and growth patterns. Fleming shares his findings with colleagues, presenting his research at conferences and publishing papers. His discovery generates significant interest within the scientific community. However, many remain skeptical about the potential of penicillin as a therapeutic agent. Despite the initial excitement, challenges lie ahead. Early experiments and challenges, a long road ahead. Undeterred, Fleming continues his research. He experiments with different methods for extracting and purifying penicillin, but progress is slow. He collaborates with other scientists seeking their expertise. Together, they explore various avenues to overcome the challenges of mass production and stability. Fleming even tests penicillin on himself. Encouraged by the results, he begins to envision a future where penicillin could revolutionize the treatment of infectious diseases. However, despite his best efforts, Fleming faces an uphill battle. Funding for his research is limited, and the technical challenges remain formidable. Despite the setbacks, Fleming never gives up hope. The year is 1939. World War II engulfs Europe. Amidst the turmoil, a beacon of hope emerges from an unlikely source, a dusty research paper. The paper, authored by Alexander Fleming a decade earlier, details the discovery of penicillin. At Oxford University, Howard Florey and Ernst Chain stumble upon Fleming's paper. Intrigued by the potential of penicillin, they embark on a quest to unlock its secrets. They assemble a team of talented researchers. The Oxford team faces an uphill battle. Penicillin, in its crude form, is notoriously unstable and difficult to produce in large quantities. Undeterred, Florey and Chain push forward with their research. They secure funding, establish a dedicated laboratory, and work tirelessly to overcome the technical challenges. As the war rages on, the need for effective treatments for battlefield injuries becomes increasingly urgent. Infections, once a death sentence for countless soldiers, now stand a chance of being cured. Penicillin, with its remarkable ability to combat bacterial infections, emerges as a beacon of hope on the battlefield. The Oxford team scales up their efforts. They conduct clinical trials on a small group of patients suffering from life-threatening infections. The results are nothing short of miraculous. Patients who would have succumbed to their illnesses just a few years earlier make remarkable recoveries. News of penicillin's success spreads rapidly. The potential to save countless lives on the battlefield drives governments to invest heavily in penicillin production. The race to mass produce this miracle drug becomes a top priority. In the United States, the War Production Board mobilizes the nation's industrial might. Pharmaceutical companies, research institutions, and government agencies join forces to mass-produce penicillin. Scientists and engineers work tirelessly, optimizing production methods and building large-scale fermentation facilities. The quest to produce penicillin becomes a national priority. New strains of penicillin mold capable of producing higher yields are discovered and cultivated. Deep tank fermentation is adapted for penicillin production. By 1944, enough penicillin is being produced to treat Allied soldiers wounded on the battlefields of Europe. Penicillin becomes a symbol of hope and a testament to the power of scientific collaboration. Penicillin's triumph. A medical revolution unfolds. Penicillin transitions from a wartime secret to a medical marvel. Mass production techniques paved the way for widespread distribution. Penicillin becomes the first widely available antibiotic. Its impact on human health is immeasurable. Diseases like pneumonia and meningitis become treatable. Surgical procedures become safer and more routine. Penicillin revolutionizes healthcare, transforming treatment of bacterial infections. The success of penicillin ignites a golden age of antibiotic discovery. Scientists intensify their search for new antibiotics. New classes of antibiotics are discovered, providing a powerful arsenal. The world marvels at the transformative power of penicillin. The fear of bacterial infections begins to recede. Life expectancy increases dramatically. Penicillin marks a turning point in human history. The legacy of penicillin, a triumph of serendipity and science. The discovery of penicillin stands as a testament to the power of observation, the importance of scientific curiosity, and the unpredictable nature of discovery. Alexander Fleming's keen eye, his willingness to explore the unusual, and his meticulous record-keeping transformed a chance observation into a medical revolution. Fleming's story reminds us that breakthroughs often come from unexpected places. It highlights the importance of fostering a spirit of inquiry, of encouraging scientists to follow their curiosity wherever it may lead. The seemingly insignificant, the accidental finding, can hold the key to unlocking profound truths about the natural world. The development of penicillin into a life-saving drug is a testament to the power of collaboration, the importance of bringing together diverse minds to solve complex problems. The Oxford team, led by Howard Florey and Ernst Chain, built upon Fleming's initial discovery, demonstrating the power of teamwork and shared vision. Penicillin's legacy extends far beyond its initial impact on treating bacterial infections. It ushered in a new era in medicine, the age of antibiotics, transforming the way we approach healthcare and fundamentally altering our relationship with disease. The double-edged sword: antibiotic resistance and the future of medicine. The widespread use of penicillin and other antibiotics has undoubtedly saved countless lives and alleviated immense suffering. However, the success of antibiotics has come at a cost. The overuse and misuse of these drugs have led to the emergence of antibiotic-resistant bacteria, posing a significant threat to global health. Antibiotic resistance is a natural evolutionary process. When bacteria are exposed to antibiotics, those with mutations that confer resistance are more likely to survive and reproduce. Over time, these resistant strains can become dominant, rendering antibiotics ineffective. The rise of antibiotic resistance is a pressing global health concern. Infections that were once easily treatable are becoming increasingly difficult and, in some cases, impossible to cure. The World Health Organization has identified antibiotic resistance as one of the biggest threats to global health, food security, and development. Addressing antibiotic resistance requires a multi-pronged approach. We need to develop new antibiotics, reduce unnecessary antibiotic use in both healthcare and agriculture, and promote responsible stewardship of these life-saving drugs. Beyond bacteria, penicillin's impact on scientific research. The discovery of penicillin not only revolutionized medicine, but also had a profound impact on scientific research. It spurred the development of new techniques for growing and studying microorganisms, leading to a deeper understanding of the microbial world. Scientists, inspired by penicillin's success, began to explore the potential of other natural products as sources of new drugs. Penicillin's impact extends beyond the field of medicine. Its discovery highlighted the importance of basic research, of pursuing scientific knowledge for its own sake. The story of penicillin reminds us that even seemingly esoteric research can have unexpected and far-reaching practical applications. The tools and techniques developed during the penicillin era continue to shape scientific research today. From genetic engineering to synthetic biology, scientists are building upon the legacy of penicillin, pushing the boundaries of knowledge and developing new solutions to global challenges. The discovery of penicillin serves as a powerful reminder that science is a journey of exploration, full of unexpected twists and turns. It highlights the importance of curiosity, perseverance, and the willingness to embrace the unknown. A timeless lesson: the enduring power of observation and collaboration. The story of penicillin is a reminder of human ingenuity and scientific collaboration. Fleming's legacy lives on in the countless lives saved and the lessons his story teaches. It is a story about observation, serendipity, and the enduring human spirit. The story of penicillin serves as a beacon of hope and a reminder of the power of science and collaboration.